Our next speaker is Professor Kim Peel, who is an advanced PhD from the KAIS, London Media Biotechnology. And then in the postdoc experience, he served as a professor in Catholic University for 22 years. So not just to serve as a professor, has founded some company whose name is Hemolab. So him is already mentioned from here as well. So he has founded those company 9 years ago. Now when he met, he just called me the CEO, not a professor anymore. So no. Right, so today he will give us Sony will give us some plant-based one, and Professor Joe was mentioned about touching some cultivating it, and then Professor Kim, maybe he will just give us some other fermented-based one. So let's give him applause for his talk.
Thank you for your introduction. I still am a professor now. Okay, my name is Bill Kim, and I am working at the Catholic University and also leader of novel food ingredient for alternative proteins and founder of Hemolab, former CEO. So I'm a professor now. So what I'm going to talk about today is a story how we select a rapid dividing cell and it derived cell protein induce multiple life form to be healthy, and how would we achieve further to improve some special nutrient. So these days people are grocery shopping and they worry about the nutrition, not only nutrition fact, they also worry about sustainability, global warming, infectious disease, and the origin of the food, non-GM food or ultra-processed food, something, very much. Cell-derived protein can solve many questions. So, next slide. Okay. So cell-derived protein or single-cell protein refers to protein or culture's biomass of the culture's edible cell. And unlike conventional proteins from field for livestock farming or fishery or plantation, cell-derived protein is a fermentation process and happens indoors, so it's not affected by weather, and many factors can be intentionally controlled during the manufacturing, as Sunil said. So we have hypothesized that if among these many edible cells, we have hypothesized that if you have a rapid dividing cell, it would certainly have. Sorry. Bad boy. It would certainly have some micronutrient enriched in their cells to support their own growth. So it could be very nutritious to another life forms. So we began to select a rapid dividing cell by using a continuous culture with gradually increased washout conditions. So it worked like 1,000 hours, and you get a rapid dividing cell eventually. And among the cells remained not be washed out, we picked up the colonies and manufactured to cell protein like this, composed of over 50% protein content of biomass. And later on, I'm going to further talk about how we further improve the special target nutrient later on slide. But anyway, we feed this to animal test. When we feed this to chicken, the weight gain was not good because they consumed less. We thought it was not tasteful to chicken anyway. But the feed efficiency was better. And when we dissect their cecum, the cecum microbiome was cecum lactic acid bacterial number was several fold higher. So we thought it is nutritious, but not tasteful to chicken. So we feed them to layer chicken for egg, and we were able to harvest a healthier and thicker shelled and darker yolk colored egg. So it's not the commercial. We made another product, dog food. It only contained cell protein 0.2%, but anyway, it was relief that dogs liked it this time, unlike chicken. And we got many reviews that the customers told us that their dog has a better stool consistency and better smell. Also, when we analyzed the fecal samples, it showed a better microbiome structure, diverse and healthier structure. So to make sure, we fed them to mice with high-fat diet in a growing stage. When you feed high-fat diet only, you get a big roundish mouse. But in the supplemental group, the cell protein supplemented group, you get still big, but looks slimmer and looks angular style. Its microbiome analysis showed us still unhealthy and diverse microbiome structure. Dissected body weight showed especially subcutaneous fat was lower, and their thigh muscle was higher. Was strange. Blood contained the lower triglyceride and cholesterol level, as well as the lower inflammation level, inflammation indicator level. So to make to understand how it worked, we repeated the same experiment again with a mouse and to collect their intestine tissues. So the experiment went well, so microbiome diversity also showed a diverse and healthy gut in an in vitro co-culture model system consisting of 1 beneficial bacteria, 4 commensal bacteria, and 5 pathogens. And we added these cell proteins. It showed only beneficial bacteria got growth, and the number of bacterial physiology showed the animal host of health favorable conditions, such as higher cholesterol degrading activity, higher bile salt hydrolysis activity, which is favorable to the host. So this protein powder helps other bacteria, especially anaerobic bacterial growth was supported, and this one was also supported on other examples, like this protein powder cultured bifidobacterium cells showed more storage time, so survived longer, and this protein powder cultured strict anaerobic bacteria, including Akkermansia, survived longer against the toxic oxygen exposure. Also, it restored the imbalance of the microbial gut like this into a normal eubiosis condition. So balance, reconstruct their microbiome structure. So we concluded this protein powder is supporting other bacterial promotion because we thought it was right. They have many nutrients that balanced to other life forms. Okay, another reason, action mode would be work on the gut epithelium cell, the goblet cell, which is present in your inside gut epithelium. And as you can see here, the protein powder fed group intestine showed more blue spots here. Is an epithelial, the goblet cells staining, so more activation, and it really showed a higher increase, several-fold higher. Depends on the location of the intestine, but anyway, it all increased the mucin layer. So if you think about it, the mucin layer is kind of a hydrophilic barrier in the gut. So it will reduce the hydrophobic lipid component, nutrition, transport into your bloodstream. So that explains why it has got lower fat accumulation compared with another. This protein didn't have control, didn't have animals. So this animal cell line supporting story is also supported another story. One of my colleagues has published that he has a pig cell line and he wants to replace the FBS. So he used this protein extract instead of the FBS. He got a cell line proliferation, and through the cell line it showed a higher energy generating signal pathway, which is the cytochrome P450, and it doesn't have a cancerous signal. A couple of days ago, he also called me. Then he wants to tell me that he was succeed on the what was that? Muscle cell line from cow. So he works on too, and I expect his publication soon. Anyway, so it's quite a supporting story. So to sum up, this vital protein, rapid growing dividing cell-derived protein, showed good for microbiome structure and bacterial physiology, especially for the anaerobic bacteria, good for animal cell line and producing more mucin, and which lead to host animals' health as one synergistic health action. So synergistically it worked together. So the reason would be it has, as I told at the first time, it has rapid dividing activity. So it should have many nutrition inside. So it contains rich micronutrients in a natural form. Second is its protein content is consisting of essential amino acid in a balanced way. Compared to soy protein or cricket, the insect protein, has got something bias in the amino acid. It has relatively fair, and also compared to other single cell protein, yeast extract, spirula or chlorella proteins, it has a better balanced amino acid form. Third reason can be this. It has a special internal oligosaccharide structure, which is an arabinan, galactan, lipid mannan. And this oligosaccharide can support especially lactic acid bacterial growth. So we can talk about it later. But anyway, this reason is work together as a pretty healthy food. So people commonly know, or we empirically know, that ultra-processed or purified food can cause an imbalance of the nutrition, because it should be carefully designed. Otherwise, it'll impurity, so the balance would be collapsed. So the ultra-processed or purified food can cause many health defects, such as overweight. When we add purified hemin to the mouse gut, it showed microbial diversity. Diversity was not as good as the ones from the cell proteins. The cell protein, which is micronutrients, are bound to their protein. So I think this is one of the reasons, because it's natural and already balanced. And to make sure, we add this protein powder to an imbalanced diet for purpose, like a high carb, high protein, high fat. High carb comes from many plants combination, and high protein comes from chicken breast, and high fat from pork belly. And you see several cases, but commonly they showed an increase of healthy microbiome diversity and showed an attenuate their health risk several cases in several ways. So you can see the detailed data in the poster presentation number 20. She is right there. And okay, so then I'd like to talk more about how do you emphasize then. It was not tasteful to chicken. Dogs like though. Chicken doesn't like it. I don't know if human would like it or not. So how do you emphasize a special nutrient target component? We have designed our target nutrient cell selection method, which named the GATE. It's not important, but anyway, it consists. We constructed a plasmid DNA system consisting of a promoter responding to intracellular target metabolite, and that linked to a gene of growth promotion genes. The growth promotion gene can be several, like an energy generation enhancer or a translation fitness enhancer. Okay, in short time. Well, we'll talk about it later, but anyway, it's got published already. So this plasmid harboring cell is designed to sync the target metabolite concentration and growth rate. So this host can be selected, naturally selected in a continuous culture too. So once you remove the selection plasmid out, you have a non-GMO cell showing on higher contents, higher target metabolite. So we were able to select an heme-emphasized Corynebacterium glutamicum cell, which gives you a more meaty taste. Glutamate-emphasized C. glutamicum, which gives you more umami taste. And we also have succeeded on the yeast strain, Saccharomyces cerevisiae, so different hosts work in this way. So the rapid growing selection strategy was working fine, but if you emphasize one target nutrient, this one would be a good way to go. Okay, let's wrap up. So we selected rapid dividing cell-derived protein, and it promotes many life forms, beneficial bacteria, anaerobic bacteria, animal cell cultures too, and it produces mucin promotion to the host and attenuate many health risks from the imbalanced diet in force. So it could be good for MediFood for patient or some precision nutrition for dieter or senior or athlete, as well as the alternative protein consumers. So I appreciate that is all. I appreciate for the colleagues and organizer and for the funding. Thank you for your listening. Thank you.
Okay, we have another 5 minutes, so maybe it's better 2 or 3 questions. Is that acceptable? Then I will give you some questions. So actually, for everybody knows to reaching out the goals for the commercializations.
Yes.
There's 2 factors: qualifications and affordability. So you already reaching out to that goal because you just produce some single cell. It's not a big deal. You're already doing some very well within the many company. So for qualification, you already have a very precisely some data set for the some animal test. But I'm just curious about among your mentions. So chicken doesn't like it, and dog like it. So how we can make it some human like it?
So that's why I showed the last slide. How do you emphasize a target? But I think that would sacrifice some another health effect derived from that margin increase of the specific target, because the rapid dividing cell means it's evolutionary adapt to the cells, so just fit for their own growth, and that is nutritious to other cells. So I'm not sure, but well, for human, yes, we can manage to, and also we have another way to masking the flavor or texture. Actually, I don't know that much.
So the reason is why asking for the Son is products because expert from the experience with the Impossible Food, right? So plant-based meat is quite suffer from the vinegar odors and something like that, and the Impossible make it more tastier.
Yeah,
So,
yeah, that is possible.
so, yeah.
Thank you. When you compare the like the nutrient-enriched single cell or microbial cell approach, whether compared to purified micronutrients, do you have data about the cost structure?
Cost.
Cost, because you avoid the.
I'm sorry, no,
the
but
purification step,
but I'd like to make this way unpurified, because the last slide, this one, the purification is fine, but if you add intentionally add to food to make delicious, tasteful, I think that'll make some imbalance to your food. So I'd like to make it natural, but and I haven't done that cost evaluation, no, but it's better this way.
Okay, any other questions? Oh, yeah, close one.
3 questions.
So you didn't show the data, but I'm sure you must have included the negative control as the normal Corynebacterium extract.
Yes, I did.
The impact of adding the nutrient enriched.
I did it for the, what was that? Animal cell line proliferation test. Yes, I did, and not my colleague did, actually. Yes, I did. I didn't do that for the animals test because it costs you a lot. But yes, in a lab scale test, yes, I did.
So the difference is?
Yeah.
All right, let's give him a big applause again.
Okay.
Yeah, we